Hello and good afternoon friends, welcome to the CEC-UGC live lecture. Dear friends, we are pleased to announce that we have started with a very new series on genetics, and so far we have conducted some sessions for you. In continuation with those sessions, today also we are going to study a very new topic under genetics. Today we would be going to study on modifications of Mendel's monohybrid ratio, and for this we have once again with us within our studios Dr. Varunendra Singh Rawat. Dr. Rawat is Assistant Professor in Department of Zoology, Hindu College, University of Delhi. So dear friends, let's welcome our guest Dr. Varunendra Singh Rawat, and under his able guidance, let's understand more and more about genetics. So without wasting any time, I would like to welcome our guest Dr. Rawat. Dr. Rawat, welcome to the UGC lecture.
Thank you. Today I'll be talking about modifications of Mendel's monohybrid ratio. As I've already talked in previous lectures, how monohybrid ratio is arrived at using the experiments of Mendel. Mendel was also known as the father of genetics, and using his conducting various experiments, he came to certain conclusions about inheritance. In today's lectures, I'll be talking how a monohybrid cross is done and how a monohybrid ratio is achieved using Mendel's law. Also, I'll be talking about various modifications which a monohybrid ratio of Mendel's undergo due to various changes in gene expression. So the learning objective for today's lecture is various modifications of Mendel's monohybrid ratio. Before I talk about various modifications of this ratio, I would like to go through what exactly is monohybrid ratio using the example of Mendel's experiment. Mendel did monohybrid cross, wherein he took a particular character and he used purebred lines of a given character which was giving alternate phenotype. So in this particular example, as we see, there are two different strains, two different lines, one of which gives yellow seeds which is coded by dominant allele capital Y, and the other is giving green seeds which is coded by recessive alleles small y. When these two lines are crossbred or when they are cross-fertilized, it leads to formation of individuals which are having allelic combination capital Y and small y. This particular combination leads to formation of yellow colored seeds because of the dominance and recessive relationship between these two alleles. Capital Y is dominant to small y, so it will be able to express its phenotype, whereas small y can express its phenotype only when it is present in homozygous condition, that is small y. So F1 individuals, when it is cross-fertilized with other F1 individuals or when it is self-fertilized, it leads to formation of two types of gametes. The male gametes will be capital Y and small y. Similarly, the female gametes will also be capital Y and small y. When these gametes fuse together, they give rise to individuals which are genotypically either capital Y or capital Y and small y. The capital Y individuals will be bearing yellow seeds, and the capital Y small y individuals will also be bearing yellow seeds, whereas individuals which have small y will be green seeded individuals. So the genotypic ratio in this particular cross is 1:2:1. 1 is for capital Y, 2 is for capital Y, small y, and 1 is for small y. Whereas the phenotypic ratio for this particular cross is 3:1. There are three yellow individuals, yellow seeded individuals, and one individual which will be producing green seed. So this is a normal Mendel monohybrid cross ratio for genotype as well as phenotype. In a particular case study, it was found out that the flower color in 4 o'clock plant, also known as Mirabilis jalapa plant, and also in snapdragon plant, it was seen that when purebred lines which produced flower of red colors were crossed with purebred lines which produced flower of white colors, they lead to formation of F1 individuals which produced pink colored flowers. The plants of F1 generation are having pink color in spite of the parents being red and white. So, the normal Mendelian monohybrid cross suggests that if there is dominance and recessive relationship between the two alleles, then this particular individual or F1 individual should be either red color or it should be white color. But in this particular case, it is leading to formation of individuals which are producing pink colored flowers. These F1 individuals, when they are self-fertilized, they lead to F2 individuals which have genotypic ratio of 1:2:1, which is the normal Mendelian genotypic ratio. In this particular case, CR, CW and CW are the genotypes, whereas phenotypic ratio is 1:2:1 for red colored flowers, pink colored flowers and white colored flowers, whereas in a normal Mendelian cross it should be 3:1 phenotypic ratio. So, this is in contrast to Mendel's monohybrid phenotypic ratio of 3:1. This particular case shows incomplete or partial dominance. Whenever a cross between parents with contrasting traits may generate an intermediate phenotype, then this particular relation is known as partial dominance relationship. Herein, genotypic ratio is identical to Mendelian monohybrid cross ratio, but neither allele is dominant, so the phenotypic ratio is identical to genotypic ratio, which is 1:2:1. Another example of incomplete or partial dominance can be seen in foot feather in pigeons. Now, most pigeons have scales on their feet. They do not have any feather because the ancestors also had scales. However, there are some pigeons who have feathers on their legs and feet. The extent of feather can vary. There can be just a hint of fuzz on the feet, or there might be small feathers. And then there are cases where feathering is extreme, so that these feathers almost look like wings. The foot feather phenotype in pigeons is controlled by slipper gene, which comes in two forms or two alleles, slipper allele and no slipper allele. The slipper allele is partially dominant to no slipper allele, which signifies that whenever slipper allele and no slipper allele are together, then both of them will code for an intermediate phenotype. Whereas when slipper allele is present in homozygous condition, it will code for its specific phenotype, and no slipper allele in its homozygous condition will code for some other phenotype. So, in this particular case, whenever slipper allele is present in homozygous condition, the individual, these pigeons, they have feathers in foot which are very long. They almost appear like mini wings. Whereas, whenever there is an individual which has a copy of slipper allele and a copy of no slipper allele, then it leads to formation of individuals whose foot have miniature feathers. On the other side, whenever the individual has homozygous condition of no slipper allele, then foot do not have any feather. It has only scales. So, this particular foot feather condition can be represented as FS, whereas mild foot feather can be represented as FS, and no feather can be represented as FNS. So, this particular example is also that of partial dominance or incomplete dominance, where both the alleles are expressing themselves and a mixed or an intermediate phenotype is getting produced because of this particular combination. In humans, incomplete dominance can be seen in hair type. In humans, straight hair is coded by H1 allele of H gene, that is coding for the texture of the hair. So, individuals who are having H1, that is a homozygous condition of H1 allele, they will be having straight hair, whereas those individuals who have homozygous condition of H2 allele, that is H2 in double dose, they will be having curly hair. Wavy hair is coded by individuals who have one copy of H1 allele and another copy of H2 allele. So, the parents which are H1, when a particular parent who is H1 mates with a parent who is H2. The individuals which are born in F1 generation are H1. These individuals, when breed among themselves or crossbreed, they lead to formation of individuals which will be having either straight hair or wavy hair or curly hair. The straight hair individuals will be in ratio 1, whereas the wavy hair ratio individuals will be in ratio 2 and curly hair individuals will be in ratio 1. So, in this particular case, genotypic ratio as well as phenotypic ratio is similar, that is 1:2:1. Thus, it denotes an example of incomplete dominance in Homo sapiens. Another prominent case which comes into study in genetics very often is that of Mn blood group in humans. Carl Landsteiner and Philip Levine discovered a glycoprotein on the surface of red blood cell. In humans, two forms of this glycoprotein exist, which are designated as M form and N form. The glycoprotein can be either M glycoprotein or it can be N glycoprotein. An individual may exhibit either one or both of them in his or her red blood cell. It is not dependent on sex of the individual. The Mn genes are located on chromosome number 4, and this particular locus has two alleles which are designated as Lm and Ln. The genotype LM codes for M glycoprotein on the RBC of individuals. Genotype LN codes for Mn glycoproteins, that is both M and N glycoproteins are present on RBC of the individual, and genotype LN codes for N glycoprotein on RBC of individual. So, any individual who is having Mn phenotype or alleles LM-LN, when he or she mates with individuals who are having Lm and Ln alleles or Mn blood group, it will give rise to individuals who are either M blood group or M phenotype having alleles Lm-Lm, or individuals who are having N blood group or Ln-Ln alleles, and individuals who are having Mn blood group or Lm-Ln alleles. And these are produced in ratio of 1:4 for M phenotype, 1:4 for N phenotype, and 1:2 for Mn phenotype. In this particular example, we can see that there is expression which is characterized by a distinct expression of both the alleles which are present in the individual. If the two alleles, namely M and N, are present in the same individuals, both will be expressed on the RBC as glycoprotein. That is, there is no mixing or production of an intermediate phenotype. So, these products of the alleles are phenotypically detectable, there is no intermixing. And this particular pattern is known as codominance, because both the genes or both the alleles for a given locus can express themselves when they are present together. One example of codominance is seen in chickens, and it basically relates to feathers in chicken. The white feathers in chickens are coded by alleles FW, whereas black feathers are coded by alleles FP. When any individuals having genotypic combination FW is mated with individual having genotypic combination FB, that is white feathered cock is mated with a black feathered hen, then individuals which are speckled might result. These speckled individuals have alternate feathers of black and white color because they have in their genotype a combination of alleles at this particular locus, and they have FW and FB alleles. So, both the alleles are able to express themselves producing black feathers and white feathers simultaneously, leading to formation of an individual who is speckled in appearance. So, it can be summarized here that FW is coding for white phenotype in feathers, FB is coding for black phenotype in feathers, FB is coding for black and white phenotype in feathers. So, individuals who are FB when mated with individuals who again are FB or those who are speckled individuals, they will lead to offsprings who are FW or white feathered, FB or black feathered, and FB or speckled feather, and the ratio for these will be 1/4 for white, 1/4 for black, and 1/2 for speckled. This again is an example of codominance. Now, there can occur cases where a particular locus has more than 3 alleles. This particular combination might be having dominance recessive relationship for various alleles, or it might be having specific expression so that all the alleles are getting simultaneously expressed, that is codominance, or it might be having expression pattern where there is intermediate phenotype is resulting when more than one type of allele are present in the individual. So, multiple alleles are those alleles of a given locus whose number is more than 3 in the population. These particular alleles can only be studied in populations because any individual can have maximum of 2 alleles for a given locus if that individual is not having any extra chromosome in his or her genotype. So, normally all individuals have only 2 alleles for a given locus. Members can also have alternate forms of gene, that means any individual can have any form of the allele, but the restricting condition is that one individual can have only 2 types of alleles or 2 alleles at a given time. So, inheritance of ABO blood group in humans, which was studied by Karl Landsteiner, established that these particular alleles are they can be categorized as multiple alleles. These blood groups are characterized by presence of specific antigens on the surface of RBC. The locus for ABO blood group is at the chromosome number 9. Different blood groups are inherited due to presence of 3 alleles, namely IA, IB, IO, which can also be named as small I, in different combinations. I stands for isoagglutinogen. IA is responsible for production of A antigen. IB is responsible for production of B antigen, whereas IO or I does not produce A or B antigen. That means whenever individual is having double copy of IO or I, he or she will not be producing either of the A or B antigen. So, this particular figure shows how different genotypes are formed in individuals and what particular blood type they will result in. So, any individual who is having 2 copies of IA will be A blood type, any individual who is having 2 copies of IB will be B blood type, individuals who are having 1 copy of IA and 1 copy of IB will be AB blood type, and those individuals who are having 2 copies of small I will be O blood type, whereas individuals who are having 1 copy of IA and 1 copy of small I will be A blood type, and those who are having 1 copy of IB and 1 copy of small I will be B blood type. These particular glycoproteins are formed from a precursor molecule which is known as H antigen. This H antigen or H substance is the precursor to A and B antigen. H substance is formed when a fucose group is added to an already existing oligosaccharide on the surface of red blood cell. This particular oligosaccharide is made up of a unit of glucose, galactose, N-acetyl glucosamine and galactose each. When fucose is added to this particular combination, then H antigen is formed. A antigen is derived from H antigen when N-acetyl glucosamine is added to the H substance. So, whenever there is addition of N-acetyl galactosamine to H substance, A antigen is formed on the surface of RBC. When a galactose unit is added to the substance, this particular H substance, then it leads to formation of B antigen on the surface of RBC. However, when this substance stays as such, that is, there is no modification by addition of N-acetyl galactosamine or addition of galactose, then it leads to formation of individuals who are having O blood type or O blood phenotype. This particular figure shows that individuals who are having O blood group, they do not have any addition of extra carbohydrate to the already existing H substance, whereas B blood group individuals have galactose added to the H substance, A blood group individuals have N-acetyl galactosamine added to the H substance, whereas AB blood group individuals have both N-acetyl galactosamine and galactose added to the H substance. So, individuals who have these AB genes, they will be expressing both A glycoprotein and B glycoprotein on their surface. Another example of multiple alleles was given for RH blood group system. RH stands for rhesus monkey. There were two different classifications for RH blood group system. One was given by Alexander Wiener and another was given by Ronald Fisher and Robert Race. Alexander Wiener suggested that at a particular locus there are multiple alleles for RH blood group, and these alleles can be put in as R1, R2, RO, RZ, small r, R prime, R double prime and RY. Whereas Ronald Fisher and Robert Race believed that instead of only one gene, there are three closely linked genes present in the given loci, and each of them had the capability of producing a specific antigen. And each of the given gene had two alleles, namely capital C, small c for C locus, capital D, small d for D locus and capital E, small e for E locus. However, it was suggested that the D allele, capital D allele encodes the most significant antigen, which leads to formation of RH type individual, that is RH positive individual. So, the individuals who are having capital D in their genotype will be coding for RH antigen on their RBCs. This particular table sums up the categorization of Wiener genotype and Fisher-Race genotype and phenotype. So, individuals who are having R1, R2, R0 or RZ according to Wiener genotype will be RH positive, whereas those individuals who are having at least one copy of capital D will be RH positive according to Fisher and Race genotype, whereas the other genotypes according to Wiener, which are R, R prime, R double prime or RY, and individuals according to Fisher Race genotyping who do not have a single copy of capital D gene, they will be termed as RH negative because they will not present any RH antigen on the surface of their RBC. Another common example of multiple allelism can be seen in ducks. It is seen in plumage in ducks, that is the wings, the feathers of ducks. For this particular locus, the coloring of the feathers is controlled by three alleles, namely MR, M and MD. MR is dominant in relation to M, which is dominant in relation to MD. So, there can be three phenotypes, namely restricted mallard, wherein only the extremities of the wings have the white color, whereas a normal mallard has white in a significant proportion. This is a normal wild type individual, whereas Duffy individual does not have white coloration, it is brownish in appearance because the feathers are brown. Whenever any restricted individual is mated with a mallard who is M and MD and restricted is capital MR and MD, the individuals can be formed either of restricted mallard phenotype or mallard phenotype or dusky phenotype. There will be individuals, half of the individuals will be restricted mallard phenotypically and 1/4 will be mallard, whereas 1/4 will be dusky individuals. Whereas genotype for the restricted mallard can either be MRM or MRMD, and genotype for mallard is M and MD, and for dusky it is MDMD. So, the progeny is half restricted phenotypically, half mallard and half dusky. Another example of multiple allelism can be seen in white eye locus in Drosophila. It is a recessive I mutation which was discovered by Thomas Hunt Morgan and Calvin Bridges in the year 1912. More than 100 alleles are present at this locus and they lead to formation of eye color which ranges from white to brick red wild type. There are various alleles at this particular locus, like W leads to white, pure white color, WA leads to yellowish orange color, WSAT leads to deep ruby color and WBF leads to light buff color. These are the different phenotypes of eye color in Drosophila, and these are caused because of a number of alleles present at the eye color locus of Drosophila. Coat color in rabbit is another example of multiple allelism, wherein C plus leads to formation of wild brown fur in rabbit, CCH leads to formation of black tipped white fur, CH leads to formation of white fur with black paws, nose and ear and tail, and C leads to formation of albino, which is a white colored individual. C plus is dominant to CCH and CCH is dominant to CH, which is dominant to C. These are the different genotypic combinations which can lead to specific phenotypes in the rabbit, and CC is the recessive homozygous individual which leads to formation of albino color. Another example, another type of modification can be seen in lethal alleles. Here mutation leads to production of alleles which might code for non-functional gene product. These recessive lethal allele will express only in homozygous state, but in some cases a recessive lethal allele also produces a distinct phenotype in heterozygous state. Herein we can see that yellow coat color is coded by AY, which is dominant to A gene. So any individual who is having AY is yellow coat colored, individual who is having AYA is also yellow coat colored, whereas individual who is AA will be agouti coat colored. It is seen that when yellow individuals are mated among themselves, they lead to formation of individuals who are either capital Y, small y, small y, small y, or capital Y, capital Y. It is seen that this particular alleles act as recessive homozygous for lethality, and all the individuals who are having capital Y genotypic combination will die because it acts as a recessive homozygous lethal, whereas individuals who are capital Y and small y will be yellow colored, and those who are small y will be non-yellow colored. All the individuals who are capital Y will die in embryonic stage only. So we will get 2 by 3 of the progeny who are yellow and 1 by 3 of the progeny which are non-yellow. Many such examples of lethal alleles exist, like curly wing in Drosophila, plum eye in Drosophila, dicot wing in Drosophila, stubble bristles in Drosophila, and lyra wing in Drosophila. These are the modifications of Mendel's monohybrid ratio. Thank you. Today I will be talking about modifications of Mendel's dihybrid ratio. Mendel was a great scientist who is also known as father of genetics. He performed certain experiments which established rules for monohybrid crosses as well as dihybrid crosses. Today I will be discussing various modifications of Mendel's dihybrid ratio. Before that, I will be talking about how a dihybrid cross is made and what exactly is a Mendel's dihybrid ratio. After that, I will be talking about interaction of genes among themselves which brings about modifications of this particular Mendel's dihybrid ratio. So, the learning objective for today's lecture will be about modifications of Mendel's dihybrid ratio. A normal Mendel dihybrid cross involves breeding of two different purebred lines which breed true for their particular character. At a particular time in both the lines, two characters are taken, like for example in this particular case the roundness and the color of the seed is taken at a given time, that is simultaneously. So, if a plant which is grown from a round and yellow pea is crossbred with a plant which is grown from a wrinkled and green pea, it will lead to formation of F1 individual who will be having round and yellow pea, because capital R is dominant to small r and capital Y is dominant to small y. R codes for yellow seed color and capital R code for yellow seed color, and capital Y codes for yellow seed color and capital R code for round seed, whereas small r codes for wrinkled seed and small y code for green color. So, the F1 individuals will be capital R, small r, capital Y, small y, and all the seeds will be expressing the dominant trait which is coded by capital R and capital Y. When the plants having this particular genotype are crossbred, or if there is self-fertilization among these particular plants, individuals of 4 different types can be gained. They can either be round yellow, round green, wrinkled yellow or wrinkled green. Round yellow phenotype, round green phenotype, wrinkled yellow phenotype and wrinkled green phenotype will occur in ratio of 9:3:3:1 because of various genotypic combination of alleles. So, a normal dihybrid cross will result in individuals who have phenotypic ratio of 9:3:3:1, whereas genotypic ratio is different. A dihybrid cross can be ascertained from the fact that the F2 phenotypic ratio is always expressed in 16. So, in the previous example, the phenotypes were in ratio 9:16, 3:16, 3:16 and 1:16. So, any cross which shows phenotype in the ratios of 16 will be a dihybrid cross. In a Mendel dihybrid cross, the principle of segregation and independent assortment are followed. The laws established by Mendel that the alleles segregate independently, and that when we are considering two different locus, then the alleles at two different locus are assorting independent of each other. These particular laws are followed during dihybrid cross. However, many cases have been studied and seen wherein there is interaction between genes, so that a normal Mendelian ratio, phenotypic ratio in F2 progeny is not seen. This might occur because several genes might influence a particular character, that is, one character might not be expressed by only one gene, it might be influenced by more than one gene, or cellular function of numerous gene products contributes to the development of a common phenotype. Though they might not be directly leading to formation of phenotype, there might be certain function of other gene products which can lead to contribution of a common phenotype, and therein lies the reason where because of which there is change in the normal Mendelian dihybrid ratio of 9:3:3:1. One such interaction is known as epistasis. Epistasis is it occurs when effect of one gene or gene pair masks or modifies the effect of another gene or gene pair. The alleles at the masking locus are epistatic to those at the second locus. Also, alleles at the second locus which are getting masked by those at the first locus, these alleles are known as hypostatic, whereas those which are causing masking, these alleles are known as that particular locus is known as epistatic locus. Now, recessive epistasis is again a condition wherein recessive homozygous condition at one locus masks the effect of dominant alleles at other locus. An example is that of coat color in mice. In mice, agouti coat coloration is dominant to black coat coloration. Agouti is basically white hair with black tips, which leads to formation of greyish phenotype. This particular agouti color results from genotype A or A small a, whereas black coloration results only when there is recessive homozygous condition, that is small a. At the second locus is present another allele wherein presence of recessive homozygous condition, that is presence of small b will eliminate pigmentation completely from the mice regardless of allele at locus A. This leads to formation of individuals which have albino phenotype. Therefore, this suggests that for pigmentation to occur, at least one dominant allele should be present at second locus, that is B locus, for the mice to appear agouti or black. So, individuals who are agouti, when they are bred with individuals who are albino, that is P1 generation is agouti and albino, it leads to formation of F1 individuals who are again agouti with one copy of each dominant and recessive gene, recessive allele for both the locus. When F1 individuals are crossed, it leads to formation of individuals which are having specific genotype and specific phenotype. F2 individuals are crossed when capital A, F2 individuals are formed when capital A, capital B and small b individual is again crossed with individual who is capital A, capital B, small b. And as can be seen here, individuals result who are either agouti or albino or black in coloration. Different genotypic combination give different coloration. Albino is formed whenever there is a recessive homozygous condition at B locus, whereas black coloration results whenever there is either single or double copy of dominant B allele at B locus, and at A locus there is recessive homozygous condition. So, the individuals which result are agouti 9, black 3 and albino 4. Agouti individuals to black individuals to albino individuals are formed in ratio of 9:3:4. These are again represented in ratio of 16, which ascertains that though there are only 3 phenotypes, this is a dihybrid cross which has become modified because of this particular specific interaction which is known as recessive epistatic interaction. This particular interaction results because the precursor molecule for pigmentation is acted upon by the product of dominant B gene. When this particular gene product or B enzyme acts on the precursor molecule, it leads to formation of a pigment which is black in color. This black pigment is then acted upon by the product of capital A allele, which leads to formation of agouti pattern. If at B locus there is small b, that is there is no dominant allele, then there will not be any formation of black pigment, and even if at A locus capital A allele is present, agouti pattern will not result. So, the down steps are not taking place if there is small b condition in the genotype of individual. So, the individual will be albino. So, this particular type of modification which leads to formation of 9:3:4 ratio is known as recessive epistasis condition. A peculiar case of blood group inheritance was seen in Mumbai, and it was thereafter named as Bombay phenotype. It was discovered by Dr. Y. M. Bhinde, and later on it was found that in human beings only about 4 individuals per million can be seen with this particular phenotype. So, this particular Bombay phenotype was found out when a woman who was having O blood group was admitted to the hospital, and when her genealogy was constructed, it was seen that her parents, the father was having A blood group and the mother was having AB blood group, and her brothers were having AB and B blood group. So, this particular woman genotypically cannot be O blood type, because she might inherit either A allele or B allele from her parents, and she should be either A type or AB type or B type. But this woman was O blood group. She was married to an individual who was having A blood type, and their progeny were having either AB blood group, A blood group or B blood group. This is also not possible, because since this woman is O blood group, the individuals, the offspring which are born can only be A blood type or O blood type. Now, how does this particular condition occur? This occurs again because of recessive epistasis. Recessive homozygous condition at one locus in human beings is masking the effect of dominant alleles at other locus, in this particular case A and B allele. Individuals who are small h genotypically, they will be phenotypically O blood group even if they are having A or B allele at second locus. This occurs because H antigen gene in dominant condition, it encodes an enzyme known as FUT1 enzyme, which synthesizes H antigen. This particular locus is present on chromosome 19. However, if H allele is present in recessive condition, that is small h, then FUT1 enzyme is not produced, an inactive form of that particular enzyme is produced, so that H antigen cannot be synthesized in these individuals. So, individual who is having small h at H locus will be producing, not be producing a normal H antigen. So, even if that individual is having A or B genes in its genotype, there will not be formation of any A antigen or B antigen. So, phenotypically that individual will be O blood group, because RBC will not be carrying glycoprotein A or glycoprotein B, it will be carrying the incomplete H substance. So, later on it was ascertained that it is indeed the case, this woman is having one allele which is IB, which will be coding for B blood group, but because of presence of small h in H locus, this individual is not able to form H substance, and consequently B antigen cannot be formed. So, this individual was not having B blood type, instead she was having O blood type. So, this particular phenomenon is known as Bombay phenotype because of this particular discovery in Mumbai. Another type of modification is dominant epistasis. Dominant epistasis occurs when a dominant allele at one locus masks the effect of alleles at other locus. This particular modification can be explained with the help of summer squash, which is a fruit, and fruit color in this particular vegetable, it is a vegetable, can be explained because of dominant epistatic interaction. Herein, dominant allele at A locus results in white fruit color. If at A locus there is recessive homozygous condition, then the fruit color is yellow. However, there is a condition, it becomes yellow only if at the other locus there is presence of at least one dominant B allele. So, if there is capital B condition or capital B small b condition, and at A locus there is small a condition, then the fruit color will be yellow. However, if there is small a condition at A locus and at B locus there is recessive homozygous condition, that is small b, then the fruit color will be green. So, individuals who are white in color, when they are mated with then which are green in color, that is capital A, capital B is mated with small a, small b, then F1 individuals are formed which are capital A, capital B, small b, and these individuals are white in color. When these individuals are crossbred, it leads to formation of F2 progeny. And as can be seen from the given table, 3 type of phenotypes result, which can be either white or yellow or green. Yellow individuals result only when there is at least one copy of dominant B along with recessive homozygous condition at A locus, small a, small a. Green individuals result when there is recessive homozygous condition at B locus as well as recessive homozygous condition at A locus. All the other individuals which are having at least one copy of capital A will produce white individual, because this is dominant epistasis. Dominant A allele will not let the pigmentation or color formation to take place in the summer squash fruit. So, the ratio for different phenotype is 12:3:1. There will be 12 individuals who are white, the ratio will be 3 for yellow and 1 for green. Still there are only 3 phenotypes, but we can ascertain that it is a modification of Mendelian dihybrid cross, because the phenotypes are present in the ratio of 16. Another modification which has been studied is that of complementary action of genes which are present at different locus. Herein, presence of at least one dominant allele of each of the 2 gene pairs is essential for expression of a particular phenotype. At least one copy of dominant allele at both the locus is required for expression of a given phenotype in this particular modification. An example is that of flower color in sweet peas. In sweet peas, if there is at least one copy of capital A and one copy of capital B, it will lead to formation of purple color flower. That means simultaneously there has to be present at least one copy of A and B. So, individuals or the lines which were white in color, because of presence of only one type of alleles, capital A in dominant condition and small b in recessive condition, and another line which is small a and capital B, both will be leading to formation of white color. When they are bred, they lead to formation of F1 individuals who are purple in color, because they have one capital A and one capital B individual. These individuals, when crossbred, they lead to formation of F2 progeny, which can either be purple or white, which again depends on presence of the allele. All the genotypes where there is only one type of dominant allele present, they will be white, whereas all the others which have at least one copy of each of the dominant allele will be purple in color. So, we get a ratio, phenotypic ratio of F2 as 9:7, 9 purple and 7 white flowers, which again is in ratio of 16. We can also check for complementation from the given cross. If 2 white lines are bred together and they only lead to formation of white lines, then it suggests that there is no complementation. That means the mutation is occurring at the same locus. The alleles are of the same locus. Whereas, if there is formation of purple color flower after mating between 2 white colored lines, it means complementation occurs, and in both the lines, each of the lines, mutation is occurring at separate locus. To explain this, we can use this particular figure wherein M1 is coding for uncoordinated behavior in mice, M2 is again coding for uncoordinated behavior. If a wild type allele is present along with M1 and M2, it leads to formation of wild type individual who is normal. If these individuals are crossbred and we get individuals who again show uncoordinated behavior even when there is presence of M1 and M2 alleles in the individual, it means that there is no complementation and both the mutations are alleles of the same gene. However, if after crossbreeding there is formation of individuals who are wild type, that means they do not show uncoordinated behavior, it means complementation has occurred and both the mutation were present on alleles at separate locus. So, this is a complementation analysis which helps in finding out about mutations at different locuses. So, the complementary action of genes comes into place wherein a precursor molecule is changed by product of one of the allele, and the intermediate product which is thus formed is acted upon by product of another allele, which leads to formation of final product. So, if any of the allele in dominant condition is not present, then there won't be formation of the final product, which is purple pigment, as seen in the example of sweet pea. Another modification is that of duplicate, which can be termed as duplicate dominant genes. Herein, a character can be brought out by one of the two dominant alleles at two separate locus, except for the recessive homozygous condition, which produces a different phenotype. The phenotype coded by dominant gene will be produced in all the other cases, because both the alleles at both the locus can lead to formation of the same phenotype. This can be seen in Shepherd's purse. Shepherd's purse is a plant which produces a capsule which can be either triangular or ovoid. Triangular capsule results when there is present capital A and capital B in either single copy or double copy, and ovoid purse results when these particular alleles are present in recessive homozygous condition. These individuals, when crossbred, lead to formation of F1 individuals who are triangular, and F1 individuals, when again interbred, they lead to formation of F2 individuals, of which all except one is triangular, and the one which is ovoid is having double recessive homozygous condition, small a, small b. So, the ratio final phenotypic ratios for duplicate dominant gene is 15:1. The triangular Shepherd's purse is present in ratio of 15, whereas ovoid Shepherd's purse is present in ratio of 1. So, there is again 16 ratio, though only two phenotypes are present. So, it is a modification of Mendelian dihybrid cross. So, duplicate dominant genes are basically working on the same precursor molecule, and they are leading to formation of the same phenotype. That is, there are two pathways for production of the same phenotype. Another modification is that of suppression, wherein an allele at one locus suppresses the effect of gene at the second locus. The recessive homozygous condition of one allele facilitates the production of other phenotype. Recessive homozygosity at both locus produce phenotype which is similar to that produced due to suppression. For example, we can see here, if a colorless individual is a colorless individual of primula which produces a colorless flower, that is white flower, is again mated with another purebred line which again produces white colored flower, though the genotypes are different, capital A, capital B and small a, small b, they will lead to individuals who are all colorless in F1 progeny. However, these individuals, when they are mated among themselves or crossbred, they lead to formation of individuals who are either blue, producing blue colored flowers or white colored flowers. So, the primula individuals will be producing either white flower or blue flowers in ratio 13:3, and this is because of suppression effect. Now, suppression is occurring because the precursor molecule is getting converted into blue pigment because of action by product of gene A, capital A. So, if capital A is present and in locus B it is recessive homozygous condition, then individual will be colored. However, if capital A is not present and in B locus it is capital B, then the individual will be colorless, and if at locus A there is recessive homozygous condition, then the individual will again be colorless. However, this capital B interferes with the production of B pigment. So, even if capital A is present and there is capital B at locus B, that is dominant allele B is present at locus B, there won't be any formation of blue pigment and the flowers will be colorless. So, this is an example of suppression. Now, another example is that of duplicate genes with cumulative effect. When dominant alleles at both the locus produce a specific phenotype, this is known as cumulative effect. The dominant alleles at only one locus produce a different phenotype, and double recessive homozygous condition produces a third phenotype. It can be seen in the shape of the summer squash, the fruit of the summer squash. If there is capital A, capital B individual, it will be disc shaped whereas individuals who are small a, small b, they will be long shaped. When these individuals are crossbred, they will lead to formation of individuals who are capital A, small a and capital B, small b, producing disc shaped individual. When these F1 individuals are crossbred, they will lead to formation of specific types or specific phenotype, and one more phenotype appears, which is spherical phenotype. So, we will be getting in F2 generation either disc shaped individuals, sphere shaped individuals or long individuals. So, here it can be seen that the ratio for the disc shaped individuals, sphere shaped individuals and long shaped individuals is 9:6:1, that is again in the ratio of 16, and this occurs because one gene when present in dominant condition, it is leading to one phenotype, but when at both the locus the genes are present in dominant condition, they are leading to different phenotype. In this particular case, presence of only one dominant allele at one locus leads to spherical phenotype, whereas if at both the locus at least one copy of dominant allele is present, it leads to disc shaped phenotype, and in recessive homozygous condition it leads to formation of long phenotype. Similar is the case with coat color in Duroc Jersey pigs. Again they are formed in the phenotypic ratio of 9:6:1, because whenever at both the locus there is present at least one copy of dominant allele, red colored pigs are formed. If only one copy of dominant allele is present at one locus, it leads to formation of pigs who are sandy in appearance. Whereas if there is double recessive homozygous condition, that is small r, small s, then individuals are formed who are white or albino in coloration. This is basically the effect of duplicate genes with cumulative effect. This is all about modifications of Mendelian dihybrid ratio. Thank you.
With this note, thank you sir, thank you so very much for giving us a very resourceful content on genetics. Dear friends, we believe that you might be benefiting from these lectures. If you want to give your feedback regarding this particular lecture or the lectures pertaining to genetics, then you can mail us at info.cc@nic.in, as well as if you have any kind of queries or questions to be asked, then also you can mail us. We would love to solve your queries next time when Dr. Varunendra Singh Rawat visits our studio, and for accessing this lecture the number of times you wanted, very soon we are going to upload these lectures on YouTube. You can see these lectures, you can later on give your feedback. So we would be meeting again very soon. Till then, take care, goodbye. Thank you sir, thank you so very much.